Less than 1% of drugs that enter a laboratory ever reach a patient. Not 1 in 10, not 1 in 50, 1 in 100, at best. Every failed molecule absorbs years of labor, millions of dollars, and frequently the last realistic hope for patients waiting on treatments that never arrive. The pharmaceutical industry has operated on this arithmetic for decades, absorbing losses, distributing costs forward, and calling the outcome acceptable because occasionally, against long odds, something works. In October 2025, Eli Lilly announced it would build a machine designed to break that equation. 4 months later, in Indianapolis, it turned on. The system is called LillyPod. It delivers more than 9,000 petaflops of AI computing performance through 1016 NVIDIA Blackwell Ultra GPUs. It holds access to approximately 700 terabytes of genomics data. It is the most powerful AI supercomputer wholly owned and operated by any pharmaceutical company on Earth. Traditional pharmaceutical laboratories test around 2,000 molecular hypotheses per year. LillyPod runs billions of simulations simultaneously. This is not a faster version of the existing process. It is a structurally different process, and it has already begun searching for drugs that have never been imagined. Drug discovery, across most of its modern history, has been a science of elimination conducted at human speed. A research team identifies a biological target, typically a protein implicated in a disease mechanism. Candidate molecules are synthesized, screened against that target, evaluated for binding stability, and discarded when they fail. The process is methodical, expensive, and constrained by human intuition at every decision point. The failure statistics are severe. Oak Ridge National Laboratory documented that fewer than 1% of drug candidates beginning in the laboratory ever reach the market. That figure does not reflect negligence. It reflects genuine biological complexity. A molecule that performs precisely in isolation can fail catastrophically in a living system, triggering off-target effects, metabolizing incorrectly, or simply failing to reach the intended tissue. The financial consequence of that failure rate compounds across the full pipeline. The Tufts Center for the Study of Drug Development estimated in 2016 that bringing a single new drug to market costs approximately $2.6 billion. The majority of that figure is not the cost of the drug that succeeded. It is the aggregated cost of all the candidates that failed, redistributed onto the one that survived. Timeline compounds the problem further. From initial discovery to FDA approval, the average drug requires 10 to 15 years. A compound identified as promising in 2025 reaches patients in 2035 under favorable conditions, which, statistically, almost never hold. The screening bottleneck anchors the problem. Human researchers working in physical laboratories test roughly 2,000 molecular hypotheses per year. High-throughput screening systems introduced in the 1990s pushed that number upward, but the underlying constraint persisted. Molecules had to be physically synthesized and physically tested before anything could be concluded about them. Computational chemistry has been eroding that barrier for decades, but remained limited by available processing power. Simulating a single high-fidelity protein-molecule interaction at sufficient resolution can consume hours on conventional server infrastructure. Scaling that to millions of simultaneous evaluations was beyond reach, until the hardware caught up. In February 2026, in Indianapolis, Indiana, Eli Lilly activated a system with no direct precedent in the pharmaceutical industry. The announcement came in October 2025. Lilly disclosed it was constructing LillyPod on NVIDIA's DGX SuperPod architecture, specifically using DGX B300 systems, making it the first deployment of that configuration anywhere in the world. From announcement to activation, approximately 4 months. A project of this infrastructure scale would have required years a decade earlier. Lilly completed it in a single fiscal quarter, LillyPod is wholly owned and operated by Eli Lilly. That ownership structure carries deliberate strategic weight. Pharmaceutical companies have previously accessed high-performance computing through partnerships with national laboratories or shared cloud infrastructure. By owning the system internally, Lilly ensures that the proprietary genomics datasets, unpublished molecular research, and active clinical development data flowing through the machine never leave the company's control. The hardware defines the machine's operational scale. LillyPod runs 1016 NVIDIA Blackwell Ultra GPUs, delivering more than 9,000 petaflops of AI performance. The system holds over 290 terabytes of GPU memory, which determines the size and complexity of models that can run in active operation. Accessible genomics data reaches approximately 700 terabytes, a catalog of genetic variation, protein structure records, and molecular interaction data large enough to ground biological predictions in population-scale reality. Alongside the hardware, Lilly established a co-innovation lab with NVIDIA. In that structure, new AI capabilities are developed, validated, and deployed into the system continuously. LillyPod does not carry a fixed capability profile. The models running on it improve over time as research advances. Lilly describes the result explicitly as a continuously learning system, a drug discovery engine that grows more capable with each deployment cycle. The claim that LillyPod runs billions of molecular simulations simultaneously requires precise definition because the word simulation carries specific technical weight in this context. In traditional drug discovery, a molecular simulation models the physical interaction between a candidate compound and a biological target, how a molecule docks with a protein, what structural changes result, whether the binding is stable enough to produce a therapeutic effect. At the resolution needed to distinguish between two molecules differing by a single atom, that calculation is computationally intensive. A single high-fidelity simulation can demand hours on standard server infrastructure. What LillyPod's architecture enables is not simply running one simulation faster. It runs vast numbers of simulations in parallel across a computational space that spans proteins, small molecules, and genomic data simultaneously. The 9,000 plus petaflops of AI performance are distributed across concurrent workloads, not concentrated on sequential tasks. The system can evaluate a small molecule candidate against a protein target, while simultaneously training a genomics model that might identify an entirely new target. The foundation model approach underpins this capability. LillyPod trains what researchers call foundation models, large AI systems built on broad biological data, then adapted for specific tasks. This is the same architectural logic behind large language models in text processing applied to molecular biology. A foundation model trained across protein structure data does not require rebuilding when a new protein target is identified. The accumulated biological knowledge transfers. Lilly's stated scope covers proteins, small molecules, and genomics, three distinct biological data types processed within a single integrated system. The 700 terabytes of genomics data available to that system provides essential grounding. Raw computational throughput without high-quality biological training data produces unreliable outputs. The genomics archive gives LillyPod's models a reference library of human biological variation substantial enough to make its predictions physically meaningful. Human scientific expertise is powerful and structurally bounded. A researcher working a specific disease pathway develops genuine depth over years of focused work. That expertise is also constrained by the literature absorbed, the experiments conducted, the molecular families considered plausible, and the assumptions embedded in the field's shared training. These are not individual failures. They are inherent features of how human knowledge accumulates. AI systems trained on broad biological datasets do not share those constraints in the same configuration. population. A foundation model trained simultaneously across proteins, small molecules, and genomic data can identify statistical patterns spanning biological scales that no single researcher would naturally connect. A genomic variant associated with disease resistance in one population might correlate structurally with a protein conformation exploitable by a small molecule, a connection requiring a researcher to hold thousands of variables in simultaneous consideration. For a model processing 700 terabytes of genomics data against a molecular structure library, that correlation surfaces as a pattern in training. This is the specific capability Lilly is targeting with LillyPod's multi-domain foundation model architecture. By training AI systems across multiple biological data types simultaneously, the system can surface drug candidates outside the solution space a human team would explore. It can identify molecules operating through mechanisms not yet named in the scientific literature because naming follows observation, and observation at this scale requires instruments no human team can replicate manually. The implications extend to target identification, which many researchers consider the hardest problem in drug discovery. Most pharmaceutical programs begin with a known biological target, but the mechanisms underlying most diseases involve networks of interaction, feedback dynamics, and emergent effects that only become visible when analyzed across large data sets. A system with access to population-scale genomics data can, in principle, identify disease mechanisms that existing research has not yet characterized, generating new targets rather than only better candidates for established ones. Lilly has not disclosed specific novel targets identified by LillyPod since its February 2026 activation. Drug discovery timelines mean that a compound identified in the system's first operational months would not produce a clinical candidate for years, but the architecture capable of generating those discoveries is now running. The clearest available evidence for what supercomputing does to pharmaceutical timelines comes not from LillyPod directly, but from a partnership that preceded it by 2 years. In 2024, BPG-BIO announced an exclusive partnership with Oak Ridge National Laboratory's Frontier Supercomputer. Frontier operates at 1.1 exaflops, more than 1 quintillion calculations per second. Oak Ridge has characterized that speed as equivalent to the entire global population working without pause for 4 years. BPG-BIO used Frontier for biological data processing tasks that had historically required 6 to 9 months. Running on Frontier's infrastructure, those tasks completed in approximately 9 hours. The time reduction exceeds 99%. That compression does not translate uniformly across the full drug development pipeline. Phase 3 clinical trials require years of patient data, regardless of how fast the underlying molecular modeling runs. Regulatory review timelines are set by statute and institutional process, not computing throughput. But the discovery and preclinical phases, which historically account for several years of the total development timeline, are precisely where supercomputing exerts direct force. When a process that currently requires 9 months in the discovery phase can be completed in 9 hours, the practical effect is not only speed, it is optionality. A research team operating on 9-hour cycles can test more hypotheses, pursue more leads simultaneously, and abandon failing candidates before multi-year sunk costs accumulate. The economics of failure change when failure is identified earlier. LillyPod's 9,000+ petaflops of AI performance and Frontier's 1.1 exaflops of general high-performance computing are optimized for different workload types and cannot be compared on raw numbers alone. But the directional logic of BPG-BIO's results applies across both systems. When the time cost of a discovery phase computation drops by multiple orders of magnitude, the strategy and economics of the research program change at the structural level. Lilly spent approximately $9 billion on research and development in 2024 and maintained over 60 molecules in clinical development as of 2026. LillyPod represents a specific calculation that a measurable portion of that expenditure is currently absorbed by the inefficiency of the discovery process. New episodes publish every week. Subscribe so the next one reaches you the moment it lands. The pharmaceutical industry has absorbed and discarded major research paradigms before. Combinatorial chemistry in the 1990s was expected to eliminate the discovery bottleneck by generating massive candidate libraries. It produced data volumes that overwhelmed analytical capacity and largely failed to improve success rates. Genomics, after the Human Genome Project completed in 2003, was expected to unlock disease mechanisms broadly. It revealed biological complexity that took 2 additional decades to begin navigating effectively. LillyPod is operating in a different environment than either of those moments. The AI architectures running on the system are not speculative frameworks. They belong to the same class of foundation models that have already demonstrated measurable performance in protein structure prediction, genomic analysis, and molecular property forecasting. DeepMind's AlphaFold 2, released in 2020, predicted 3-dimensional protein structures with accuracy matching experimental methods developed over decades. That capability, considered implausible before 2018, is now standard input for the work LillyPod is designed to extend. The continuously learning architecture is the feature that most distinguishes LillyPod from a conventional high-performance computing installation. A standard supercomputer carries a fixed capability profile that ages with its hardware. LillyPod's co-innovation framework with NVIDIA means new AI models are developed, validated, and deployed into the system on a rolling basis. The machine running in late 2027 will carry materially greater capability than the one activated in February 2026, not through hardware replacement, but through model improvement accumulating across deployment cycles. Lilly has committed to operating LillyPod on carbon-neutral, fully renewable energy by 2030. That timeline indicates the company treats the system as permanent infrastructure, not a pilot program with a planning horizon extending at least 4 years from activation. The competitive pressure this creates across the industry is direct. Pfizer, AstraZeneca, Roche and Novartis each maintain substantial computational biology operations. None currently operates a dedicated AI supercomputer at LillyPod's stated scale. That gap either closes as competitors build comparable systems, or it widens as LillyPod's continuously accumulating model improvements compound Lilly's advantage year over year. The 1 in 100 drug failure rate does not dissolve because a faster system enters operation. Biology retains its complexity. Trials retain their duration. Regulatory review retains its requirements. But the discovery end of the pipeline, where failures are identified earliest and at lowest cost, is where LillyPod concentrates its force. The machine does not need to solve biology. It needs to be better than the process it replaced. By that measure, the threshold has already been crossed. The pharmaceutical industry's core constraint has never been a lack of scientific ideas. It has been the time required to test them, and the cost of discovering, years too late, that a candidate was wrong. Fewer than 1% of drug candidates that enter a laboratory reach a patient. Every year of delay in the discovery phase is a year added to the weight at the other end. LillyPod, 1,016 GPUs, 9,000 petaflops, 700 terabytes of genomics data running in Indianapolis is Eli Lilly's answer to that constraint. The machine is operating now. The results will take years to reach clinics, but the search, at a scale no prior laboratory could sustain, has already begun. Subscribe to follow what comes next.